matter what you start with, there's going to be the capacity to use isomerase and reductase enzymes to get to the point where you can feed into beta-oxidation. And if we see a structure of a compound and it's already partly oxidized, you should be aware that you can't get as much energy out of it.
And so the whole process is just to get that double bond between 3 and 2?
Yeah.
And that's the only way that it can get the energy from it?
Well, it's the only way that the hydration enzyme works.
Okay.
So you can't go forward, in beta-oxidation unless you have the double bond positioned here with respect to the esterified CoA. Because that's the binding determinant, the recognition determinant for the enzyme that adds the water across here, which is the next step that you have to do. You can't bypass that step. You need that structure. So all these convolutions are about getting you to here. Right? If you have one problem, you kind of get it out of the way, and you can do one round, and you got another problem, you got to deal with that. This thing might have 4 double bonds. You consecutively have problem after problem. Just deal with it, and the cell can deal with it. Okay, so what about odd numbers of carbons? So you could have an odd number with double bonds, so make your life as maximally difficult as possible if you're the cell, or require the most number of new activities. But let's just say that we got through all that stuff and the double bonds, or else we just didn't have any to begin with. We're going to end up with a 3-carbon compound. If we had C13 or 15 or 17, right? That's what we're going to end up with, propynyl-CoA. And so then what do we do? So again, the cell has enzymes to deal with it. The first enzyme is the carboxylase. It's a biotin-dependent enzyme. It actually goes from 3 carbons to 4. Okay. And so that uses up an ATP because you're forming a carbon-carbon bond. It's an energy-requiring reaction. So biotin is a 1-carbon carrier. Okay. I think we saw the structure of biotin in the previous reaction. We didn't really do the mechanism so much, but at least you know that biotin is a kind of cofactor that transfers 1 carbon units. And then we have an epimerase, which is a rearrangement enzyme. Right? And we saw that in the context of sugars. We had epimerase sugars in one of the sugar entry pathways where we were moving around the hydroxyl groups on the carbons from one chirality to the other at particular positions. Right? So here what we're doing is this epimerase is basically just interchanging the substituent, this substituent with the carbon in this one. It's basically taking this chiral center and moving this carboxylate to the bottom, and this group here with the CoA linked thioesterified onto it is coming up here. Right? So it's an epimerase is an enzyme, is a type of rearrangement enzyme that works on a chiral center and changes the chirality of that center. Right? Just like changing OH to NH positions on the carbon of a sugar. All right, and so then we're here, methyl malonyl-CoA, and then we're going to generate a 4-carbon compound that is suitable not for further beta-oxidation, but rather to enter the TCA cycle. Succinyl-CoA you will recognize as a TCA cycle intermediate, right? But it's halfway through the cycle, and so you feed this into here, and even though you started out with more carbons than say one where you just had acetyl-CoA, you had to invest ATP here, right? And then you got to a 4-carbon compound, but you got into one that's later in the TCA cycle, so it's not as good as putting it in as acetyl-CoA. So you end up getting less energy when you work through this way. Okay. Now this enzyme is a mutase, so again it's a rearrangement enzyme in that same general class. And now what's going on is that this hydrogen and this group are getting swapped in position so that you can get to succinyl-CoA. So how does this enzyme work? So this enzyme is a coenzyme B12 enzyme. B12 is also called cobalamin. It's one of your essential B vitamins, and it's fairly monstrous to look at. So this is it here. And this is the 5' deoxyadenosine linked cobalamin, which is the active moiety in the transformation we're talking about. But the B12 itself is just this part. This cobalt is not linked to 5'-deoxy-A. Okay. So you have this crazy corin ring system with a cobalt in the middle. I mean, this looks, you know, imagine it starts to look a little bit like a heme, but there's quite a lot of differences from a heme, and not to mention it has a cobalt in the middle. So it's got 4 ligations in the plane, and 1 is going to this group, which is always part of the cofactor, dimethylbenzimidazole ribonucleotide, and then linked to 5'-deoxyadenosine on top. How this gets formed is that there's a reaction between the cobalt center and the 5'-methylene that bridges the triphosphate of ATP to split off the entire triphosphate linkage to form this link. Okay. The details of how this happens are not really very well understood, but we do know that this is the structure of the cofactor, the derivatized cofactor that then works in the reaction. This is it might be the only known, or at least well-characterized example in biochemistry where you have an ATP molecule and you actually split off the entire triphosphate. And we're used to seeing, we looked back in chapter 13 at possible reactions on ATP, but they certainly didn't involve anything like this. And then going on from here, this is not quite right, I realized. It says consumes 1 additional ATP. Usually when we talk about consuming ATP, we mean it's used in every cycle of the reaction. It's not, it's only used 1 time to set up the cofactor. And then the cofactor on the enzyme that's doing this, which is this enzyme, you need an ATP to generate the active form of this coenzyme, but then this transformation as a multiple turnover reaction does not take an ATP. So if we were doing ATP arithmetic on this, we wouldn't assume that this takes an ATP. Right? So this is a little bit, I shouldn't have written it that way, it's a little misleading. That make sense? Okay. So this is a free radical reaction mechanism then. We're not going to worry about the details of it, but it's good to see. Here's the cobalt linked deoxyadenosine coenzyme B12. And the way this proceeds is that there's a homolytic bond cleavage, meaning that 1 electron is left here and 1 electron is left here. And so we actually generate a free radical. Right, so that's the first step. Having set up this cofactor, the first thing that happens is that this bond is broken. It's not a very strong bond, apparently, between cobalt and carbon, like that. And then we have the substrate here. So since we have a radical, right, the radical can abstract the proton from the substrate, and that generates a substrate radical. Right? Remember that the substrate is this guy, right? So all we're doing is taking this and turning it into this, and so HX means H and this group. Right? So this little X here is the same as that. Right? So we have a substrate radical, a rearrangement of the substrate radical, because when you have a radical like this, it's easy for it to rearrange. Another abstraction of a proton back from this deoxyadenosine to regenerate that radical, release of the product, which is now rearranged from what it was from here to here. HX went to XH. Here, reformation of the coenzyme, and around you go. Right? So these free radicals are abstracting protons, rearranging position. They're not exchanging hydrogen with the solvent, though. So it has to be enough sequestered from solvent so that you don't end up with water interfering.
Can you say the importance of that again? That it's not?
It's not by solvent, because if you had water nearby, you would probably end up exchanging hydrogen with the water. And if it resulted in the same product, so you just subtracted H, then that might be okay. But if you generated a reactive HO radical in the process, then that would not be a happy thing.
Is this? I'm trying to think where this is all happening again.
So this is all happening in the mitochondria, in the oxidation of the odd-numbered carbon fatty acid. So yeah, we kind of got into this mechanism here. But you have a C17 fatty acid. You run it through 6 cycles or 7 cycles. 6 cycles of the beta-oxidation, we end up with this propionyl-CoA. Okay. And then we run this pathway to get to succinyl-CoA so that this can go through a TCA cycle. This guy is basically toast as far as the beta-oxidation goes. There's no way to turn him into acetyl-CoA so that acetyl-CoA can go into the TCA cycle. That's what we did before, but here you can't, there's just no enzyme system to do that. And this is what happens instead. And now, and our free radical chemistry is involved in B12 and the mechanism of this mutase. So, it's basically the only example we get all term of a free radical mechanism. And since this is a class on enzymes, and it's good to at least have a look at one. But it's not something we need to focus on in detail, or that I would ask you about in detail. Any other questions? All right. Let's move on to fatty acid biosynthesis. The first enzyme in fatty acid biosynthesis, which now is occurring in the cytosol, okay, using NADPH, which is generated, of course, from pentose phosphate pathway. So this first reaction involves this enzyme, acetyl-CoA carboxylase, which has 3 functional subunits. Right? It's got a biotin carrier protein carrying biotin. This is the structure of biotin. Right? It's got a carboxylase activity that carboxylates biotin, and it's got a trans-carboxylase activity that takes the carboxyl group on biotin and adds it to acetyl-CoA. And the important product, which is only a fatty acid biosynthesis metabolite and not involved in beta-oxidation at all, is malonyl-CoA. So, you know this